So let's start our discussion or exploration of the metabolism of nucleotides. And first thing we will start with is the formation or synthesis of nitrogenous bases. There are two types of nitrogenous bases, pyrimidines and purines. Let's start with the synthesis of purines. So first of all, the purines take their atoms from different sources. For example, from amino acids like aspartate, glycine, glutamine, then carbon dioxide, and then form formyl tetrahydrofluorate. These molecules give different components of the purines. For instance, we can say that glycine and aspartate and glutamine give the nitrogen component of the purines, and carbon dioxide give the carbon component, and also one nitrogen component is donated by formyl tetrahydrofluorate. And you can see it here, glycine giving the nitrogen component, aspartate also nitrogen component, and glutamine also nitrogen component, CO2 giving the carbon component, and formyl tetrahydrofluorate giving the nitrogen component. So as we have been talking again and again, that metabolism of one type of macromolecule is interacting with the metabolism of other type of molecules, and here you can see how amino acid metabolism is involved in the formation of nucleic acids. Other thing which we will see later on is that also, it's a nucleoside doesn't only consist of purines or nitrogen bases, it also contains the ribose sugar, and this sugar comes from the pentose phosphate pathway. Again, it's confirming what we have been saying, that different pathways are interacting with each other, giving or taking the products of each other. First of all, there is as far as the synthesis of purine is concerned, the formation of the first component is the 5-ribosyl 1-pyrophosphate, or specifically it is abbreviated as PRPP, and the enzyme involved is the PRPP synthetase, this enzyme. Of course, ATP is involved in this formation. This is a very important starting point as far as the synthesis of purines are concerned. As far as the regulation of this process is concerned, that is the regulation of the formation of PRPP is concerned, it is activated by the inorganic phosphate, and it is inhibited, of course, by the purine nucleotides. Again, that because of the feedback mechanism, a product tends to inhibit its reaction. Then there is formation of the 5-phosphoribosylamine. It's a very important rate limiting step as far as the synthesis of purine is concerned. What actually happened here is that it gives rise to the 5-phosphoribosylamine is formed, and here the phosphate group is replaced by an amide group is donated by the glutamine. In this way, the glutamine here replaces the phosphate group, giving rise to 5-phosphoribosylamine. This is, as I have told you, that this is our rate limiting step, because the regulation of the synthesis of the purine very much depends upon this reaction. Then there are inhibitors also of this reaction. Of course, I mean, some of the most important inhibitors. Whenever we are talking about inhibition of the formation of different components of the nucleotides, it means that it has some real clinical relevance. So, for instance, when we want to kill the rapidly growing cancer cells, one of the target is that somehow you inhibit the formation of these nucleotides. Now, when you have made what we say the 5-phosphoribosylamine, then you move towards different complex sort of reactions, and these complex sorts of reaction, these are almost 9 reactions. If I show you in more detail, So from 5-phosphoribosylamine, you come up here, from here till here, there are some 9 complex reaction, and you end up with very important precursor of the purine nucleotides, that is IMP, inosine 5 monophosphate. We will not go into the detail of these 9 reactions because our main focus is here how the formation of these nucleotides, that is the purines, takes place from the inosine 5 monophosphate. As I have told you that PRPP is converted to inosine monophosphate, and this inosine monophosphate is a very major precursor of the main purine nucleosides like the AMP and GMP, adenosine monophosphate and guanosine monophosphate. This pathway requires ATP, of course, and also THF, tetrahydrofluorate. I have already shown you this, that how the 9 reactions are involved which end up in formation of IMP. Then from IMP, there are 2 pathways which lead to the formation of GTP, that is, lead to the formation of GMP and AMP. Here you can see here, this is the inosine monophosphate. It can be converted into AMP, and it can also be converted into GMP, to major purine nucleosides. And you can see here that AMP synthesis requires GTP, while GMP synthesis required ATP. In addition to this, they also, for their nitrogen ring, the nitrogen component of their rings, they also require certain amino acids. For example, in the case of AMP formation, you make use of the aspartate amino acid, while in the case of GMP formation, we take this pathway makes use of the glutamine amino acid. Here again, the amino acids are being used for the donation of the, what we say, the nitrogen components. Of course, the regulation is also again feedback. The product of the reaction will inhibit its very reaction. Then of course, you have made AMP, you have made GMP, and your final step or goal is to make the diphosphates or the triphosphates of these moieties or these purine molecules. So AMP and GMP, under the action of enzymes adenyl cyclase and guanylate cyclase, they are phosphorylated to form ADP or GDP again, and these ADP and GDPs are then further phosphorylated to form finally the GTP, the corresponding GTP molecules. And in this way, the very function of these kinase molecules is not only to provide the phosphate moiety to make the triphosphates or diphosphate components, or triphosphate or diphosphate moieties of these purine molecules, but also to keep a fair balance between the dephosphorylated or phosphorylated moieties.